Hello and welcome, dear colleagues, to my contribution. It's about the phylogenetic history and global population dynamics of wheat powdery mildew. I want to start with a question, though, first, which is, what is sustainable agriculture and why would we care about it? Well, I think it's important that the tools we developed are used wisely because it's a continuously changing and dynamic pathogen population adapting to those tools. For example, the major R gene resistances that we employ in wheat against powdery mildew diseases are a threat to break down rapidly because the pathogen population of Plumeria graminis tridici, which is the causal agent of powdery mildew, is a really dynamic and rapidly changing population. Thus, it's important, I think, to understand the population dynamics of such populations to form more holistic approaches to disease management and to lead into a more sustainable agriculture practice. Today, I want to show you how we inferred phylogenetic history and population genetic structure using three methods, one being the maximum likelihood phylogenetic tree inferences, two, principal components analyses on the genetic data, and three, being clustering algorithms using different methods. And I would say, jumping right into it, we look at the phylogenetic tree overlaid with the clustering algorithm output as rings outside the tree. We see that the phylogenetic tree is largely concurrent with the structure the algorithms determined. And so we inferred that this data set is structured into 5 larger subpopulations, being first, Turkey and other countries of the Fertile Crescent and Asia continent, second, Europe, as well as thirdly, Brazil and South Africa as a third and fourth distinct cluster. And lastly, the fifth cluster would be US, Australia, and Canada. Looking further deeper into that phylogeny, you see that Turkey formed a genetic crossroads showing relatedness to all of those other isolates of the Fertile Crescent Asia cluster, whereas Brazil and South Africa stood out as genetically distinct populations. And they clustered apparently further away from all those of the US cluster and the European cluster, which we see later in the 3D PCA plot better. We see that the isolates from the US, Canada, and Australia cluster together, which I think indicates that they share some ancestry with each other. While interestingly, all the European isolates cluster together too. I mean, it was expected in a way, just by distance. However, when we look at the membership assignment with the clustering algorithm LNPD, having 5 possible clusters here, we see that there is a pattern of mixture between European isolates and isolates from Turkey, as well as isolates from China and the US. So we infer that the European samples share some common history with those populations, or there is recent admixture going on. Then, viewing the 3-dimensional principal components plot of the data, we basically could explore another dimension of the genetic structure. And you see here really clearly how Brazil and South Africa cluster far away from the rest of the population sample, which indicates that they are really, really distinct populations sharing really little genetic similarity with the rest. And looking at this, it led us into thinking about future research. For example, we would like to determine the patterns, but also the rates of migration between those subpopulations to infer and gain more knowledge about the dynamics of this population, with hope to help tailor and select resistance genes for country-specific breeding programs for what I said earlier, a holistic approach to disease management and a more sustainable agricultural practice.